Hello everybody. Today we are going to study the third process, translation, that is involved in protein synthesis. Remember that we talked about replication that had to do with mitosis and meiosis, the duplication of the DNA. After that, we talked about the processes to produce a protein. These were two. First, transcription. We studied this in the previous video. And the other one is going to be translation. And after this process, the protein is already done, already made. Okay? So the first thing is that we finished the first process, transcription, when we synthesized the messenger RNA. If you didn't understand that process, it's much better to go back to the previous video and watch it again until you know, because if not, this is going to be a little mess. So we have a messenger RNA, and inside eukaryotic cells, we are talking that this messenger RNA is inside the nucleus. So the first thing that it has to do is to get it to leave the nucleus and go to the cytoplasm, where ribosomes are going to be. So this messenger RNA is going to leave the nucleus and it's going to go through the nuclear pores of photonucleares. No, it's going to go through them, and it's going to exit the nucleus. And then we are in the cytoplasm. Of course, if we are talking about prokaryotic cells, we don't have this problem, right? Because all these processes are made in the cytoplasm. Okay, so first, messenger RNA is going to leave the nucleus. Understood that, right? So we have this strand that is going to be a messenger RNA, and this strand of messenger RNA we are going to put here the nitrogenous bases. I have put some of these as an example, okay? And why did I put it 3 by 3? If you see them, I have joined 3 by 3. I will explain you why. Because when this messenger RNA is in the cytoplasm, the two subunits of the ribosomes, because the ribosomes are not the dots that we have been studying so far, now it's a little more complex structure, and it has two subunits, the small subunit and the bigger one. So the first thing that happens is that the ribosome, both subunits, is going to join the messenger RNA, and it's going to be like a reader. It's going to read all the nitrogenous bases that it's going to find. Okay, until it finds a code that says, okay, translation starts here. Which code is this? It's going to be 3 nitrogenous bases, and those 3 nitrogenous bases, we're going to call it codon. So when the ribosome finds this codon, which is G, I'm sorry, I couldn't remember. So AUG, okay, translation is going to start. It is always the same codon? Yes, it's always the same codon. The first one is always the same. AUG. In every strand of messenger RNA, there is always going to be a codon, the first one, that is going to be AUG. Okay, always. Then something happens. We join, the ribosome joins. And when we have this codon, these 3 nitrogenous bases are going to codify 1 amino acid. This codon is going to codify another amino acid, and so on. So every time that the ribosome is going through the messenger RNA, is reading the codons, the different codons, and that is how we know the amino acid that has to follow the first one. Okay? More or less. So, ribosome joint, we have the first codon, which is going to be the first amino acid. Methionine. Always the first amino acid in the protein in translation. This is not meaning that all the proteins that you have in your body, they start with the amino acid methionine. When this process is finished, there are some maturing processes, procesos de maduración of the protein, that they are going to change the protein, and in many cases they are going to remove the methionine as the first amino acid. But for this, always the first methionine, okay? So we have the ribosome and a transfer RNA, ARN transferente, always having attached an amino acid, as we studied when we explained RNAs, remember? It's going to join the ribosome. So it's going to come this way and it's going to join the ribosome. And it's going to leave the amino acid that he is carrying. It is carrying, okay? In this case, it's methionine. But how do this transfer RNA and not this one, for example, how does it know that it has to be this one? Because in the transfer RNA, you have a place with 3 nitrogenous bases that is called anticodon. And again, due to complementary bases, ¿vale? Las bases complementarias, we know that we have to leave methionine. Because if you have a codon that is AUG, which would be the complementary one? UAC, right? The complementary is that one. Here we go. So as these two are complementary, we know that this transfer RNA matches with this codon here. So we leave the methionine. That is called anticodon. I don't know if I said that before. Understood more or less how it works? So once that this transfer RNA releases the methionine, the transfer leaves and can be reused, and the ribosome moves forward to the next codon, which is CGA, ¿vale? So here we will have to have a complementary codon, that would be the anticodon. Which would it be? G, C, U. No? It matches. So if it matches, means that this transfer RNA, when the ribosome moves forward, is going to join the ribosome and is going to leave the amino acid that it's carrying. In this case, I put arginine. Okay? So we have now 2 amino acids. These 2 amino acids, they are going to join through a specific bond that is specific for amino acids. This bond is called peptide bond, ¿vale? Enlace peptídico in Spanish. So we go through all the codons, all the codons of the messenger RNA until we find something. Because a protein is not going to be eternal, right? You have to stop somewhere. For that, we have certain types of codons that are called stop codons. Okay? So when the ribosome reaches here, this codon, this is an example, UGA, for example, that means that there is no RNA transfer RNA carrying an amino acid that has an anticodon for these 3 bases. So at this point the strand is finished, ribosome separates, split, and the amino acid list, the amino acid strand releases, and there we go. There we have our our protein. Okay. So basically, this is translation. It's not simple. I understand that, but I hope that if you check all the steps little by little, they are not that difficult to understand. So check this video 2 or 3 times, maybe. I think I will put some videos about translation that I have that are more visual, and I think that you will understand it. Okay, so this is it. This is translation and how we create a protein. This protein, when it's released, it can go inside the cell. It can work inside the cell, or it can go outside the cell. Hormones, for example, many of them are proteins. So imagine that pancreas, this is a cell of the pancreas, and now it's producing insulin, and this insulin has to go to the bloodstream, for example. No, that could be a good example. And after this, what we are going to study? The genetic code. Because why do we know that AUG matches with this amino acid? Because someone that was really smart studied this and won a Nobel Prize due to this discovery. This was Severo Ochoa, the street of our school, right? And it was a really important discovery. Okay. So we will study this in the next video, because now I think that this is deep enough for you to have to study for a while. Okay? Thank you so much. Bye-bye.